Hi everyone, I'm Haley, and along with Nazira, we made this article presentation to fully summarize Article 3 and hopefully make it a little bit more understandable. So the article we're focusing on is a role for Wnt signaling and self-renewal of hematopoietic stem cells, or HSCs. So I'm going to focus on the introductions, methods, and a couple of the experiments with their corresponding figures, and then Nazira is going to pick it up and describe a few more of the experiments, their figures, and then the proposed experiment that we made together. So the first question I'm going to ask is, why should we even care? So the big issue is that there's a high prevalence of blood cancer. So you can see in the figure to the left that there are 186,400 new cases of GIST leukemia, lymphoma, and myeloma in 2021 alone. And these are often treated with bone marrow transplants. So you can see this is a huge issue. And with the bone marrow transplants, it's actually has a ton of risks associated with it. So first we're going to go through this little schematic, so it shows how it works. So they're either taken out of the blood or out of the bone marrow, and then you're going to isolate these stem cells, and you're going to reduce the volume of fluid with stem cells, cryopreserve stem cells, thaw them, dilute them, and then infuse them back into the patient. So oftentimes these patients are in chemotherapy or radiation therapy, and again, it is a good way to treat many cancers and diseases in blood using an autologous or allogeneic source. However, there are these risks of infection, anemia, graft failure, respiratory disease, pneumonia and liver dysfunction due to excess fluid, and graft versus host disease. So there are lots of issues, even though it is overall a beneficial thing. And so we want to find a better way of doing this. And so we're going to be focusing on hematopoietic stem cells, or HSCs. So we're going to be looking at this figure for a minute, and you can see that their two key characteristics are self-renewal and differentiation, specifically into these different blood cell types you can see here. Something to note, which this figure doesn't do a fantastic job of, but you can see in a couple of places, is that the nucleus actually changes depending on which cell it's in, and so it actually gets smaller. So the HSCs typically have a very high nucleus to cytoplasm ratio, while something like a red blood cell will have no nucleus. And so this is something key to note for future experiments. And then here is some of the background that the article presented. So HSCs have already been purified and characterized both phenotypically and functionally with this family of LEF1 TCF expressed. And then they have shown that Wnt signaling has affected lymphocyte progenitor cell proliferation. And so now we're going to be focusing a little bit on that Wnt signaling and beta-catenin pathway. So if you haven't watched Module 10 yet, I highly recommend doing so. However, I did pull this figure from Module 10 and we'll be going over it a little bit. So as Dr. Braffman said, you can think of it as a light switch. So when the light is turned on, you have that Wnt ligand, and it's going to bind to your receptor, and it's going to take this protein complex, and it's going to make sure that this beta-catenin is not phosphorylated. When it's in its off state, you have no Wnt. This protein complex is here, and the beta-catenin is being phosphorylated. So beta-catenin is always continuously produced and degraded in the off state. So in the on state, you're going to have this transcription of the target genes. In the off state, you're not going to have this transcription. So basically what Wnt is wingless related integration site, and they're secreted factors or glycoproteins. And then here's what I described before about them binding to the receptor to prevent phosphorylation, and that typically beta-catenin is continuously produced and degraded. So then this is made of a large family of 19 proteins, and it regulates cell growth, motility, and differentiation during embryonic development. So the study objectives are as follows. So the problem is that even though they have been purified and characterized, we do not know anything about their self-renewing growth. So our question is, how is HSC self-renewal growth regulated? Our hypothesis is that the Wnt signaling has an effect on HSC development, and then the goal is, if this hypothesis is correct, we can provide an indefinite supply of HSCs ex vivo for treatment. So the methods and techniques that the researchers utilized were actually from a previous study, so this is the abstract from that previous study. You can read it on your own time, and I will have a schematic on the next slide to kind of go more in depth with it. So basically what they did was they isolated from mouse bone marrow. They used these HSCs from H2K BCL2 transgenic mice. And it's important that they use this specific mice because they picked HSCs that will proliferate to one factor, which is the steal factor, or SLF, and that is it. You don't need a ton of different things to make it proliferate. You don't need a lot of interactions. This is just a very simple proliferation method. And then the cells were sorted using FACS analysis based off of the expression of specific markers, so c-KIT, SCA1, low levels of Thi 1.1, and lineage markers. And then we had in vitro and in vivo HSC proliferation assays. So the in vitro one used transduction of HSCs with retroviruses. Those purified HSCs were then plated between 1 to 20 cells per well, and then the cells were counted at defined intervals. So here's that schematic I promised. So here's the mouse. They took out the cells and they defined them with these different markers, and then they grouped them together as Li 5.2, Thi 1.1 low, Lin low, c-KIT plus, and SCA1. So these are going to be used in pretty much every experiment that follows. And so basically what's going to happen is this in vivo analysis of HSC function. Once these groups of cells are further isolated, they're going to be inserted into two different types of irradiated mice to test the effect of donor versus recipient. Will there be an immune response? And so the donor was Li 5.2 mice, while the recipient was Li 5.1 mice. And so these mice were bled at predetermined time intervals for analysis, and then the cells are isolated from peripheral blood, analyzed with flow cytometry after more than 10 weeks, and then real-time PCR analysis was conducted. So now we're going to get into the real meat of the study. So this is experiment 1. Does beta-catenin expression lead to self-renewal of HSCs in vitro? So I made a little schematic to try and show exactly how the cells are sorted. So you have that group of cells that I described before. And then you're going to have beta-catenin, iris GFP, and iris GFP. So iris is just internal ribosomal entrance site. So the beta-catenin one, that is going to be your experimental, while the iris GFP is your negative control. And so beta-catenin, it's going to tag cells that have activated beta-catenin with GFP, and then the iris GFP is just going to randomly select cells to put GFP on. And then both the groups are going to be sorted with FACS, so that is the fluorescence activated cell sorting. So only the ones that have the GFP expressed are going to be isolated. And then after 60 hours, both are going to be analyzed for their cell cycle. And this is the figure here. So figure 1, activated beta-catenin promotes growth of HSCs in vitro and maintains the immature phenotype of HSCs in long-term cultures. They're infected with activated beta-catenin, iris GFP, or control GFP retrovirus and subjected to cell cycle analysis after 60 hours. So we're going to go into each of these figures below after going over the cell cycle. So in this experiment, they also used Hoist staining of DNA to see when cells were actively dividing. And so you can read a little bit more about the cell cycle in this figure, but the 3 phases that they actually studied were S, G2, and M phases. So S phase is DNA replication, G2 phase is cell growth, and M phase is when the replicated DNA condenses into chromosomes. So it's going to have like double the fluorescence intensity for dividing cells because it's going to have double the DNA. So now we can go on to the actual experimentation. So we're going to have the effect of beta-catenin transduction on short-term growth characteristics, and so this is figure 1a. You can read the figure caption below each of the captions for the rest of the slides. And then for the control vector, you're going to have 34% of these cells in the S, G2, or M phases of the cell cycle, while the beta-catenin transduced cells had 58% of the cells in this S, G2, M phase. So this higher percentage just means that there's more active cell proliferation of HSCs with beta-catenin transduced cells. So then they started testing on the long-term growth characteristics. And so you have your control vector here in the light gray and your beta-catenin with the black. And the control vector showed minimal growth, and withdrawal of SLF actually led to death within 48 hours. While with beta-catenin, you had 8 to 9 population doublings with at least 100 times the number of cells inputted, and even after SLF was withdrawn, they grew for another 4 weeks and survived for as long as 8 weeks. So you can see that they had much more cell growth with the beta-catenin than with just the normal without the activation. Now we're going to test the morphology, and this is where the cell cycle analysis, this is where that HSC part really comes into play. So we're going to look at the control vector here. So you can see that the myelomonocytic appearance is starting to occur, so they're beginning to lose their nucleus. So this is what I was talking about when I was talking about that figure before. They're starting to differentiate. They're losing their nucleus, they're becoming a different type of cell, while these beta-catenin transduced cells, they still have their high nuclear to cytoplasm ratio, 65 to 75% of them. So that just means that these are showing more of the self-renewal characteristics than these ones, which are more differentiating. Last but not least, for figure 1, they tested lineage markers, and so they tested it between the control, which is the light gray, and the beta-catenin activated, which is the black. And then here was some of the data from the paragraph. So 75 to 80% positive for lineage markers in the control vector. The beta-catenin transduced cells had 5 to 10% high levels of lineage markers with the MAC1. And then there was 50% low levels of Thi 1.1, which is this middle panel. And then they said 60% were lineage negative in the right panel, and they were testing c-KIT and SCA1. So basically, the reason I have these asterisks here is that the paragraph seemed to kind of go against what the figure caption was saying, so I just wanted to make a note that this part was a little bit interesting with the variations in the article. But the main trend is accurate. Not only are they proliferating, but beta-catenin transduced cells are maintaining their morphology and markers of HSCs. So then we also have this isolation of HSCs and in vivo study. So you have that group of cells again that I talked about. You're going to use beta-catenin iris-GFP to again tag the activated cells, and iris-GFP to just tag the random ones. You're going to sort them, and after a week you're going to insert 125 of them into irradiated mice. So irradiated mice, that just means that all of their old cells were killed. You can add these then because they no longer have their own bone marrow working. Okay, and so then here is a table. So table 1, you have your control vector, and then you have your actual beta-catenin transduced HSCs. So with the control vector, you can see that there was no reconstitution. And then with your beta-catenin activated ones, for all of the cell types, there's 100% reconstitution. And the chimerism was highest for this peripheral blood cells, between 27.6 to 86.8%, and an average of 61.2. And so to measure chimerism, they did Li 5.2 divided by Li 5.1 plus Li 5.2. So then here's another figure from the study. So beta-catenin transduced HSCs led to higher chimerism levels. That's what they're showing here. And it indicated that beta-catenin promotes HSC proliferation while preventing differentiation. So then we have some supplementary experiments. So does Wnt3a expression lead to self-renewal of HSCs in vitro? So they actually use the same assay. Researchers found the similar effect with Wnt3a alone as with activated beta-catenin. So again, they're going to plate these cells per well, and then they're going to select which wells have expanded, and then they're going to inject the cells from these wells into individual mice. And again, they analyze the chimerism and the frequency of reconstitution when they had these Wnt3a cells, or activated cells. And so for almost all of them, there was 100% reconstitution, except for this one right here, which had 80%, but overall it's around the same results. So here are a couple more figures. They had much higher growth over time of Wnt3a treated cells. As you can see here, again, that is similar to the beta-catenin ones that we had seen. They had more lineage markers of unpurified. So again, the purified Wnt3a followed more closely with what HSC would look like. And then 74.8% of the cells were lineage negative, which again, that's what is typical of HSC. So it showed, again, that Wnt3a had a similar effect to activated beta-catenin on the HSC self-renewal. So from here, Nazir is going to take over, and I hope you guys enjoy. If you have any questions, please ask in the discussion.